So in this video, I want to talk a little bit about how neurons actually communicate with one another. It turns out that there's two different types of electrical signals that uh, neurons experience. They're called graded potentials and action potentials. Neurons are irritable cells. This means that they're able to receive a stimulus and convert that into an electrical impulse. And these impulses are conveyed throughout the body to cause different responses. When we consider the nervous system, we often talk about the three-neuron pathway. So in the three-neuron pathway, you have basically three neurons. Your first neuron is a sensory neuron. It's going to be the one that will receive information. It's the one that will sense a stimulus. It will convey information about that stimulus to another neuron called the integrating neuron. Now the integrating neuron is going to take that information coming from the sensory neuron, interpret it, figure out what it means, and then figure out the response to that stimulus. The third neuron in the pathway is going to be our motor neuron. It's the one that will receive a signal from the integrating neuron and then carry out the command. It will be the thing that does the response. So for example, let's say that you step on a tack. There will be sensory neurons in your foot that will recognize that you've stepped on something. Some of these neurons may even convey that what you've stepped on is very painful. That information will be transmitted to the integrating neuron as an electrical signal. The integrating neuron will receive that electrical signal, interpret it, and say, aha, I've stepped on a tack, and then it will send the appropriate signals to move your foot from the tack. So those signals to move your foot will go to a motor neuron, which is going to synapse with your muscles, and it will actually stimulate your muscles to contract and move your foot. So that's your three-neuron pathway. Now these electrical signals are of two types. You have what are called graded potentials. So these are incoming, short distance signals. You usually find them happening somewhere around the dendrites or on the cell body of a neuron. They arise from synapses with other neurons. And interestingly, these electrical signals can actually add one to another and increase, okay, the overall effect of a signal. And I'm going to talk about that a little bit later. The second type of signal that neurons use for communication is called an action potential. And action potentials are very long distance signals. They are transmitted along axons and they arise at the axon hillock. Action potentials do not summate, so that means they are not additive. Again, we'll talk about these in more detail later, but what I need you to remember is that there are two types of signals, electrical signals, that neurons use to communicate: the graded potential, which again is short distance, and the action potential, which is long distance. Now, in this video, we want to consider how your neurons are able to make any of these signals at all. So in order to do that, we've got to talk about the resting membrane potential of neurons, why it's important, and what causes it. We'll also need to talk about how you change that resting membrane potential and its relationship to graded potentials and action potentials. We're going to start by just describing the resting membrane potential, why it's important, and what causes it, aka how it's formed. When we talk about neuron communication, we usually say that neurons send messages in the form of electrical impulses or electrical signals. These impulses arise due to electrochemical gradients. Electrochemical gradient is basically a term that refers to the fact that there are differences in the concentrations of ions across the plasma membrane of a neuron. The two most important ions that you need to keep an eye on are going to be potassium and sodium. The resting membrane potential of a neuron describes a voltage that's formed because the concentration of sodium and potassium across the plasma membrane is so different. This difference is quantified as minus 70 millivolts. The minus tells you that there are more negative charges on the inside of the plasma membrane versus the outside of the plasma membrane, and the minus 70 actually comes from the ion concentrations. Now, minus 70 is actually a really big number. Most cells don't have. So the resting membrane potential is basically formed in the following way. In this figure, we have a phospholipid bilayer, so you know that's a plasma membrane. And the plasma membrane, just as a phospholipid bilayer, is impenetrable to any ions. Remember, you have those fatty acid tails, right? They don't like to mix with ions, and for that reason, ions can't cross a membrane unless you put proteins in it. So in your neurons, you have what are called leakage channels. So here are a couple of potassium leakage channels. These channels basically form a passageway for potassium to flow down its electrochemical gradient. So that means it's going to flow from areas of high potassium concentration to areas of low potassium concentration. And in addition, it's going to flow from positively charged sides of the membrane towards negatively charged sides of the membrane. Now, if a membrane, plasma membrane, only has potassium ion channels in it, okay, and usually there are a lot of these in the plasma membrane, then potassium will flow across and it will cause a negative membrane potential of 90 millivolts. Now in your neurons, potassium leakage channels aren't the only ones. We also have sodium leakage channels. So these are always open and sodium can flow across the membrane down its electrochemical gradient just like potassium. The presence of these sodium channels reduces the membrane potential, making it minus 70 millivolts instead of minus 90. The sodium-potassium pump, which you'll remember from when we were talking about muscle, is just in the membrane to maintain the concentration of the sodium and the potassium. Okay, so typically you have lots of sodium outside of the cell and only a little bit of sodium inside. And you'll have lots of potassium inside the cell and only a little sodium outside. So what the sodium-potassium pump does is it takes sodium from inside the cell and pumps it out, and it takes potassium from outside of the cell and pumps it in. So again, the overall resting membrane potential is going to be minus 70 millivolts, and this is important because if you did not have this resting membrane potential, neurons could not conduct electrical signals. They would be more like epithelial cells, which have a much, much lower RMP, resting membrane potential. So this figure is supposed to show you kind of what's going on in the neuron cell. So one thing that I hope you'll notice is that we have lots of potassium inside. The potassium is represented by these kind of salmon colored circles, and potassium can flow out just like we talked about on the last slide using these leakage channels. Now typically potassium is in a much higher concentration inside the cell, so the tendency is for potassium to flow out. So this big K here is to remind you that potassium is in a much greater concentration inside the cell than it is outside. So here's a little k outside to let you know that there is some potassium out there, but not a lot. Sodium is the reverse. Sodium is represented by these little purple spheres. So there's lots of sodium outside of the cell. Ooh, that's a kind of crazy looking N. But this is to represent the fact that there's a lot of sodium outside. And this little Na represents the fact that there's only a little bit of sodium inside. Okay, like that. Sodium can flow into the cell via its own leakage channels. Okay. Now, in addition to potassium and sodium, there are a few other players, but they're not quite as important. We have these guys, these purple spheres inside the cell. These are supposed to represent proteins, and proteins contribute a net negative charge. Proteins cannot go through the plasma membrane, so that means all of those negative charges from them are concentrated inside the cell in the ICF, and that's going to give an overall negative charge, okay, at the plasma membrane. We also have chloride ions, which are represented by these green spheres. They're very low in concentration, so again, they're not going to contribute very much in the way of charges, but they do add a little bit of negative charge to the outside of the cell. Okay. Right here at the plasma membrane, because we have all of this sodium out here, though, we're going to have a lot of positive charges. So that minus in front of the 70 for the minus 70 millivolts is supposed to let you know that the inside of the plasma membrane has a more negative charge because of these proteins and because potassium tends to flow out, okay? And the outside of the cell tends to have more of a positive charge because of the sodium outside, all right? And that's giving a positive charge. So neurons conduct electrical signals when they change this resting membrane potential. That is, when the number goes from minus 70 and becomes more positive. In the next part of this video, we want to talk about what causes changes in resting membrane potential, and we want to look at the relationship between these changes and the production of graded potentials and action potentials. So changes in your resting membrane potential occur when the concentration of ions across the membrane of the cell change and when the plasma membrane becomes more permeable to ions. So that means that more ions are able to cross. And the important thing to remember about this is that it's those changes in the membrane potential that are responsible for the graded potentials and the action potentials that neurons use to communicate with each other. Without these changes in membrane potential, neurons couldn't have those signals that allow them to receive information about stimuli, to integrate it, and then to send information about how to respond. So changes in the resting membrane potential usually happen due to the opening or closing of what are called gated channels. So let me explain what this is really quickly. Think about, for example, celebrities that live in a gated community. That basically means that when the gate to their neighborhood is closed, people can't drive cars into or out of the neighborhood, right? When the gate is open, you can freely drive into the neighborhood and drive out. With a gated ion channel, it's the same way. For these proteins, there's a little bit of protein that can actually block ions from moving across the plasma membrane when the gate is closed. When the gate is open, that little bit of protein moves out of the way and ions can cross the plasma membrane and thus change the resting membrane potential. Now there are three types of gated channels. Two of them will be especially important to our discussion today, but you need to know all three really. The first type of gated channel is called chemically gated or ligand gated. For this type of channel, it's going to open up when a specific site on the channel is bound by a neurotransmitter. So neurotransmitters are chemicals like acetylcholine that the neurons use to communicate with each other. So remember how in the last chapter we talked about on muscle cells you have a receptor for acetylcholine, and when acetylcholine binds that receptor, the channel opens up and sodium flows in and potassium flows out of the muscle cell? This is what we're talking about here. So with a chemically gated channel, some neurotransmitter binds an ion channel and then it's going to open up. Okay? We also have voltage gated channels. Okay, voltage gated channels. So voltage gated channels are going to open or close in response to changes in the membrane potential. Okay? You also have mechanically gated channels. So mechanically gated channels are channels that open up in response to deformation of a specific receptor. So for example, let's say you have pressure or tension on one of these receptors. That force actually opens up the channel so that ions can move across across the plasma membrane. And this is, just another way that neurons can use to communicate with each other. So let's talk in more detail about changing the resting membrane potential. Let's talk about the actual signals involved. So when ion channels open or close, you can get two types of signals. The first type is called a depolarization, and this is exactly like what we saw with muscle cells. We're going to decrease the membrane potential, okay, and that's going to be used for communication. The second type of change in a resting membrane potential is called a hyperpolarization. In a hyperpolarization, we're going to actually increase the membrane potential. So let me show you what that looks like. This is a graph that basically shows you membrane potential on the y-axis here and time on the x-axis. So this part here represents the resting potential. So this is for a neuron that is not transmitting any signals. It's not really receiving signals. Okay, it's at rest. Now, when a stimulus arrives at the dendrite, for example, of this neuron, and it causes some of those ion channels that we talked about to open up, let's say, for example, a sodium channel, then what happens is sodium ions start flowing into the plasma membrane, or into the cell rather, across the plasma membrane, and they're going to change the negative charges here at the plasma membrane to being more positive. As a result, this resting membrane potential, which was minus 70, okay, it becomes more positive. That is, it moves closer to 0. And we see this little bump in our graph. So in other words, the membrane potential is becoming more positive. It's getting closer to 0. And what this indicates is that the inside of that plasma membrane is becoming less negative. This is going to increase the probability that we'll get some sort of nerve impulse from this neuron. This is an example of a hyperpolarization. So in this case, we have our resting membrane potential is here at minus 70. Okay, but let's say that the presence of a neurotransmitter causes potassium to actually start flowing out of the cell. So that means the inside of this plasma membrane becomes more negative because we're losing those positive charges from potassium. Then instead of getting a bump in our graph, we get a little dip like this. So our membrane potential moves farther away from 0. It becomes more negative. And this is going to reduce the probability that we're going to produce a nerve impulse. You might think of hyperpolarizations as being an inhibitory type of signal, whereas your depolarizations are more of a excitatory or a promoting type of signal. So now that we've talked about depolarizations and hyperpolarizations and resting membrane potential, we can talk about graded potentials. So when I first talked about graded potentials, I told you that they are short-lived. Okay, these are short-lived signals, and they're going to cause localized changes in the membrane potential of a neuron. These graded potentials can be depolarizations or hyperpolarizations, and they're going to spread as local currents to change membrane potential in adjacent regions of the plasma membrane for your neuron. Now, some things that you want to remember about these as I explain them. Number one, they occur when a stimulus, it could be a neurotransmitter for example, causes gated ion channels, like those sodium channels, to open. Okay? And sometimes we're going to call these graded potentials receptor potentials or generator potentials or postsynaptic potentials. For this class, I will try to stick to using the term graded potential, but I will at some point talk about postsynaptic potentials. Just remember that those are the same thing. Something that's really neat about the graded potentials is that their magnitude, that is how big they are, is going to vary directly, okay, with how strong our stimulus is. That is, for example, how many neurotransmitters bind the ion channels and cause them to open up, okay? For this reason, because the magnitude varies with the stimulus, that's where the term graded comes from. The other thing is that these potentials do decrease in magnitude as ions flow and diffuse through leakage channels along the plasma membrane. Now, if this is hard for you to kind of visualize, don't worry. I'm going to explain it. This slide is just to kind of summarize some of the important things that I want you to remember about these graded potentials. We're going to talk about summation in another lecture, and this will make so much more sense once we get to it. So I like this slide because it kind of illustrates the principles that we talked about on the last one with these graded potentials. On this side of the slide, you have those depolarizing potentials. Remember, the depolarizing potentials get us closer to zero, and they make it more likely that we'll get an electrical impulse, aka an action potential. Over here on this side of the slide, these are all hyperpolarizing graded potentials, so remember that gets us farther away from zero. These are inhibitory signals that are less likely to give us an electrical impulse, aka action potential. So over here on the left, what they're showing you is that we have a stimulus here. It gives us a small depolarization. If we increase the stimulus, then the magnitude of that depolarization goes up and we get closer to zero. If we increase the duration of the stimulus, then the duration of the graded potential gets longer as well. And then finally, here, if the stimulus is strong enough, we might actually get a depolarization that crosses threshold and will allow us to get an action potential. Okay. So when we started talking about graded potentials, I mentioned to you that they can actually cause local currents that will spread the depolarization across adjacent membrane areas. In these next couple of slides, I want to show that to you. So what you're looking at here is a small patch of plasma membrane from a neuron, and you could think of this as being on a dendrite, for example. So here, the inside of the membrane has a lot of negative charges. Remember, this is due to the flow of potassium out of the cell, and the presence of those proteins inside the cell. If we have a stimulus, for example, let's say we've got a axon that's releasing neurotransmitter here. Oh, I shouldn't use red for that. Let me try a different color. Let's say we have an axon terminus and it's releasing neurotransmitter here. Those neurotransmitters may cross a synapse between this axon and this neuron. They may bind receptors and open ion channels. The opening of those ion channels may cause an influx of positive charges into the cell. So let's say sodium starts rushing in. The presence of the sodium changes the membrane potential so that we start accumulating positive charges here inside, and because we lost those positive charges outside of the cell, the outside becomes more negative. Now, you can kind of think of this as being a little bit like when you drop a puddle in a lake. When not a puddle in a lake, when you drop a pebble in a lake. When you drop a pebble in, what happens? You get an immediate hole, and then things kind of ripple out, right, from where the pebble went in. That's kind of what's happening here. So we had that positive charge come in, right? And positive charges are attracted to negative charges. So those positive charges start moving towards the negative charges along the plasma membrane in these directions here. Okay. Now, remember we had a negative charge outside because we lost those positive charges here. They went inside. These positive charges that are adjacent get attracted to the negative charge here. So essentially what happens is you get an influx of positive charges. Let me change colors here. You get an influx of positive charges, and then as those positive charges come in, they start moving to the side. So kind of like those ripples go out from that initial spot where the pebble went into the lake, those positive charges kind of ripple out from the initial site where you had your stimulus. Now, this is supposed to show you that as those positive charges come in, you do have little membrane channels along the membrane, and some of those positive charges will start to diffuse out. As they start diffusing out, the total charge goes down, which is what you're seeing here. Okay. And we're getting our reduction in depolarization. So this happens as we get farther away from that initial site of our stimulus, that initial site where the neurotransmitter bound those ion channels. It's for this reason that these graded potentials are considered short distance signals. The signal essentially begins here, whoops, it begins here, and then it peters out as we get farther away from that initial site of stimulus. Now, with what we've considered so far, we've looked at what happens at one location on one neuron. It's important to realize that in actuality, neurons synapse with many neurons. For example, in this figure here, you can see that each of these colored axon termini belongs to a different neuron. So if we were to look at this figure, just the top, we can see that there are synapses with 1, 2, 3, 4, 5, 6 other neurons, and that this one neuron makes synapses with 4 others from its axon. So it's like a network, really. And another thing to think about is that the signals that are coming through each of these synapses can be different. So for example, I like this figure because they highlight some synapses in green, meaning that those are excitatory signals that are coming through those synapses into this central neuron in the middle here. And then you have some synapses that are highlighted in red, which means that those are inhibitory signals coming into the neuron. So the neuron has to do a bit of math, so to speak, to add up the inhibitory versus the excitatory signals. So to add up the hyperpolarizations and the depolarizations and figure out whether or not there should be an action potential. Does the membrane potential get to a number we call threshold, which would allow an action potential to start here at the axon hillock and then move down the axon? The addition of graded potentials is called summation. There are different types of summation. The one shown here is called temporal summation. If we look at the figure on the left, we're looking at the axon termini of 3 neurons. One neuron is called A, one is called B, and then we've got C over here. These termini synapse with a neuron represented by yellow. Neurons A, B, and C, by the way, are called presynaptic neurons, and the yellow neuron here is called the postsynaptic neuron. Notice that the postsynaptic neuron has receptors. That's what these little brackets are. It has receptors for binding neurotransmitters that have been secreted by the axon termini A, B, and C. Now, for the next 3 slides, anytime you see an axon terminus that looks green, that one is secreting an excitatory neurotransmitter. That means that that axon terminus releases a neurotransmitter that causes a depolarization of the membrane for this postsynaptic neuron. Any axon terminus that appears red is secreting an inhibitory neurotransmitter that causes a hyperpolarization of the axon membrane, of the neuron membrane here, this neuron's membrane, okay, upon binding the receptor. So green means excitatory, we're going to get a depolarization. Red means inhibitory, we get hyperpolarization. All right. So in this example, axon terminus A here releases a neurotransmitter that binds the receptor here in the membrane of our postsynaptic neuron, and it causes a depolarization, which we see here. Okay, this depolarization is called an excitatory postsynaptic potential, or an EPSP. In any case, when that neurotransmitter binds its receptor, okay, it causes the depolarization and then the neurotransmitter dissipates. If another signal comes down this axon and causes the release of neurotransmitter, that neurotransmitter will bind the receptor again and we'll get a depolarization. Now, because these two events happened far enough away from each other in time, it means that we just see a little depolarization and then it dissipates. Okay. Now, if neurotransmitter is released, like we see here, and we get our initial depolarization, but before the neurotransmitter dissipates, a second dose of neurotransmitter is released, and we get a second depolarization, those two will sum together. And if we reach threshold here, we'll get an action potential like you see here. In spatial summation, we have 2 axon termini that synapse with our postsynaptic neuron, but they're very close to each other in space. So when neurotransmitter is released from the 2 neurons at approximately the same time, a graded potential gets the graded potential gets summed. So you'll recognize this figure from the last slide, right? So let's look at the right part of the slide here. Here we have 3 axon termini, A, B, and C. A and B release neurotransmitters that cause depolarization of the postsynaptic neuron's membrane. That's what these little bumps are that you see right here. C, on the other hand, causes a release of neurotransmitter from axon terminus. C, on the other hand, causes a hyperpolarization. So this is an inhibitory signal. Now, if axon termini A and B are close to each other in space and neurotransmitter is released from both at the same time, the depolarizations that we saw over here happen at the same time. They get summed together, and if we reach threshold, we get an action potential. If we look at axon termini A and C, on the other hand, if they are close together in space, okay, if the synapses from those two axon termini are close together on the membrane of the postsynaptic neuron, then when neurotransmitters released from both of those termini at the same time, we get summation, and they kind of cancel each other out. We'll talk about the types of synapses that can occur between neurons another time, but suffice it to say that two axons can synapse with each other at their termini. This allows one neuron to control how much neurotransmitter is released from another neuron. In facilitation, a synapse formed with an axon terminus producing an excitatory signal, like you see here, can cause increased release of neurotransmitter. Conversely, when there's an inhibitory axon terminus that's synapsing with another terminus, and a signal gets sent from this inhibitory axon terminus, it can actually reduce how much neurotransmitter is released from the other axon terminus. If you're looking for a place to take a quick break, now would be a good time to do it. We'll finish up this video with action potentials in just a few moments. Action potentials are a little different. Action potentials will have a brief but substantial reversal of the membrane potential, and that reversal will give us a total change in amplitude of minus 100 millivolts. This is different from those graded potentials. Graded potentials generally don't get any higher than minus 50 millivolts. Okay, so this is like twice that. You can get action potentials in the axons of neurons. They start at the axon hillock and then travel down the axon. And they also occur in muscle cells, you'll remember. Now, here's the kind of cool thing about your action potentials. Remember on the last slide how the amplitude of our depolarization basically decreased as we got farther away from that initial site of stimulus? Action potentials don't do that. They maintain their magnitude over distance, and that's why, for example, this method of communication is used for long distance. So, this is a picture of what an action potential looks like. There are 4 stages to an action potential. You have the resting state, the depolarization state, which is here, the repolarization state, and the hyperpolarization state. After hyperpolarization, the membrane potential is going to slowly return back to the resting state, and at this point, we can stimulate a new action potential. Let's look at the different stages. So when the neuron is not being stimulated, it's not communicating, that's when it's in the resting state. And as you know, the resting membrane potential is minus 70. Now, at this point, only your sodium and potassium leakage channels are open. So those are the channels, remember, that always stay open. And sodium and potassium are going to flow through those channels down their concentration gradient. In the membrane, we have some gated channels. These are sodium gated channels. And what I want you to notice here is they look a little different from those leakage channels that we looked at before. The leakage channels look like a straw. This looks like a straw, but there are actually two gates inside. Okay, you have this gate here, which controls whether ions can flow through the cell. And then you have this gate down here, the chain and ball, which is an inactivation gate. When the inactivation gate is closed, ions can't go through this protein. Okay, when that inactivation gate is open and this other gate is open as well, then ions can go through. So there are actually two gates. When a neuron becomes stimulated, let's say through the activity of neurotransmitters, local sodium channels will begin to open up, and that causes the influx of positive charges into the cell. If the stimulus is strong enough, that depolarization may reach this point, which is called threshold. So threshold is at minus 55 millivolts. This leads to a positive feedback response, and all of the sodium channels in that area will start to open up, leading to a flood of sodium into the cell, and this is going to reverse the membrane potential. So it's going to go, the membrane potential will go from minus 70 all the way up here to plus 30. This is that change of 100 millivolts that I mentioned a couple of slides ago. Ooh, that's a pretty rough looking 100 millivolts, but I want it to be there for you to remember that there is this change here. Okay, this is what's responsible for the spike of our action potential. Now, remember how I told you that those sodium channels have two gates? They have an inactivation gate and they have a regular gate that opens up. Well, shortly after the sodium channel opens, that inactivation gate will start to close. And as that happens, the permeability of the membrane to sodium goes down. So this is responsible for the next phase of our action potential, which is called repolarization. We're also going to see that potassium gates, which are in the plasma membrane and they closed during depolarization, they're going to start opening up, which allows potassium to flow out of the cell, which makes the inside of the cell, again, less positive. So here, during repolarization, we're going to see a decrease in the membrane potential, all right? And the negatively charged interior of the cell is going to get restored. So repolarization is only going to restore the electrical conditions of the neuron, but it doesn't restore them back to what we saw with a resting cell. Instead, it's the sodium-potassium pumps that are going to do that. So the activity of thousands of sodium-potassium pumps is going to restore kind of the ion concentrations for sodium and potassium in the neuron. Remember, the sodium-potassium pump pumps out 3 sodiums for every 2 potassiums that it brings in. Thus, we're going to have a rapid removal of the sodium. And remember, we just finished having an efflux of potassium, so the pumps are going to actually start bringing potassium back into the cell slowly. After repolarization, which I'll highlight here in red for you, some of your potassium pumps will actually remain open, allowing more potassium to leave the cell. This causes what's called the overshoot, or a hyperpolarization of the membrane. That means the membrane becomes even more negative than it ordinarily would be. The sodium-potassium pumps, however, will slowly add potassium back to the cell, which restores the original resting membrane potential over time. So this table basically summarizes the main points, the main differences, you might say, between your graded potentials and your action potentials. Let's talk about the graded potentials really quickly. Your graded potentials, remember, these are localized electrical impulses or electrical signals. They don't travel very far. In fact, they only travel a few millimeters. You'll typically find these originating at the dendrites or on the cell body of a neuron that's synapsing with another neuron. The channels involved will be chemically gated and sometimes mechanically gated. The amplitude of your electrical signal here is very low. So it's always less than 50 millivolts, and sometimes it's as low as around 1 millivolt. These are very fast, rapid signals. They last a few milliseconds to a few minutes. That said, they're not nearly as fast as your action potentials. Your graded potentials can be hyperpolarizing. Remember, those are inhibitory signals, or depolarizing. Remember, those are excitatory signals. And we can have summation of these signals. So that means that if we have a group of graded potentials happening, say, within short time span or within a close locations on a neuron, we can actually add those signals together, and we might be able to produce a great enough depolarization to actually stimulate an action potential. Now, let's really quickly review the action potentials. Remember that action potentials generally arise at the axon hillock. The actual area where that happens is called a trigger zone. These electrical impulses are actually generated and they are conducted over long distances. If you think about the fact that some of your neurons originate at your spine and then they go all the way down to your foot, then you know that these can be propagated over several centimeters. The channels involved here are going to be voltage gated channels. Remember, when a local depolarization is great enough, okay, we're going to depolarize the membrane to minus 50 millivolts, and that will stimulate voltage gated sodium channels to open up, giving a massive depolarization, which leads to our action potential. Action potentials are propagated again over long distances. Amplitude, I didn't talk about this. Let me talk about amplitude really quickly. All of your action potentials are going to have the same amplitude, okay? And typically that's 100 millivolts. We often describe action potentials as being all or none. That means that as long as your depolarization reaches threshold, that is minus 55 millivolts, you will get an action potential, and all of your action potentials look the same. If your depolarization is not strong enough to reach minus 55 millivolts, then there will be no action potential. So that's what we mean by all or none. You either get it if your depolarization is strong enough for threshold, or you don't get it. Your action potentials are super rapid. They are very short in duration. Typically they're from about 0.5 to 2 milliseconds long. You will have several different stages in an action potential. So you'll go from having a depolarized membrane potential to having a repolarized membrane potential to having a resting membrane potential. And there is something called a refractory period with your action potentials. So in your refractory period, you cannot stimulate a new action potential, but we'll cover that in another video. There is no summation with your action potentials. Again, they have the same size. So that is going to end this video. What are the things that I want you to remember? So for your exam, you'll want to remember a few things from this video. You should review the 3-neuron model of nervous system communication. You should compare graded potentials and action potentials in terms of their distance traveled, their amplitude, their polarity, summation, and their origin. You should also be able to, when given a diagram of an action potential, describe the stages and what's happening. For example, when is there depolarization? When is there repolarization? When are channels open? When are they closed? And during what steps? You should also generally be able to explain why the resting membrane potential for neurons is approximately minus 70 millivolts and what happens to cause that to change.